Hello students, so today in this video I'll be discussing about a very interesting topic which is very short but very interesting and very important in biological science, that is the topic of cell cycle and cell division. So cell cycle, so we know that the cell theory was modified by Rudolf Virchow, and Rudolf Virchow added another postulate to the cell theory that new cells arise from pre-existing cells. That means the earlier cell will divide to form new cells. Now when we talk about the cell cycle means how the cell passes from one division to the next division. So the entire cell cycle can be classified or can be divided and can be represented in the form of a clock where the different phases of cell cycle has been described. So the longest phase, so the first phase of the cell cycle is G1 phase. G stands for growth. So this is the first growth phase, because when a cell will proceed towards division, the cell has to collect a lot of raw materials. New organelles needs to get built up. So building up of new cell organelles and collecting the raw materials required for the cell to go into the division state, the cell passes through the first growth phase. It is the longest phase of the cell cycle. After that, the cell will enter into the S phase. S stands for synthesis. So during this phase, what happens? There is replication of DNA. The DNA amount will get doubled. The DNA will get replicated, because when a cell will divide and form the daughter cells, it cannot give away its own DNA. So it has to make a copy of its DNA. So making a copy of the DNA is occurring during the S phase. So replication of DNA occurs here. In one of my video, I will be discussing about how replication of DNA takes place. After the S phase is over, that means after the DNA has got duplicated, the cell will once again enter into the second growth phase or the G2 phase, because during the synthesis phase, during the formation of DNA, lots of raw materials gets used up. So the final growth, before the cell enters into the division state, the final growth will occur during the G2 phase. This is the second growth phase. So G1, S and G2, this all the three phases, this is also known as the I phase or the interphase. So it is sorry for the spelling, so it will be interphase. So during the interphase, what happens? The cell gets ready to go into the cell division stage. Now after this, then the cell will start dividing. So as the cell starts dividing, this phase is called the M phase or the division phase. M stands for the two types of any one of the two types of cell division, mitosis or meiosis. Now during the cell division, two things gets divided. First, the nucleus will get divided. That's why it is called, that part is called karyokinesis. Karyo means nucleus, kinesis means division. So first the nucleus will get divided, and after that the cytoplasm and other materials will get divided, and that is called cytokinesis. So in every cell division, first you will get karyokinesis, nuclear division, which is followed by cytokinesis, which is your cell division. So after the cell divides, so this is with the end of the M phase, the whole cell cycle completes. After that, the cell may go into another preparation for the next cell division. If it is a highly active and dividing cell like the meristematic tissue or the cells in a cells which are developing from the zygote, in case of blastula and all, the cells are in a state of rapid division. There the cells doesn't stop after division. They once again start getting prepared for the next round of division. However, in certain cells, like for example your nerve cells are formed only once. So once they are formed, so after that the cell remains metabolically active but does not go into preparation for the next division. So this stage of resting, it is called the G0 or the quiescent phase or the resting phase, G0 phase or the quiescent phase. So in this diagram I have shown this as the G0 phase. In some other books you may get a diagram where they might show the G0 as like this. That means after the cell division, instead of going through G1 phase, the cell enters into the resting phase. So as the cell enters into the resting phase, now that resting phase, during that resting phase, the cell will remain metabolically active. That means different types of cell cycle, different types of cellular metabolism will keep on going on in that cell, but the cell will not start collecting raw materials, or there will be no DNA replication. That means the cell will not prepare to go into the next divisional phase. Is that clear? So it is just a resting phase where the cell is metabolically active, but it is not collecting raw material to go into the next phase of division. That's why it is called the resting phase. Now this G0 phase is either absent or very short for, as I told you, for cells which are rapidly dividing in nature, for example like meristematic cells or cells of blastula. However, in nerve cells, this G0 or the quiescent phase, it is very long. Maybe the cell does not go into the G1 phase once again. It will divide just only once. So the G0 phase is very long in case of such cells. So this is the overall cell cycle occurring for any kind of cell when they go into cell division. Now we know that cell division is of two types. One is your mitosis, another one is your meiosis. So in this video I will be discussing about mitosis. In the next video I will be discussing about meiosis. So let's see what are the steps which are involved in case of your mitosis. Okay, I hope the figures are clear. Yeah. So when we talk about mitosis, mitosis is also termed as equational cell division. Now what is the reason that mitosis is called equational cell division? Because in mitosis what happens, from one parent cell, two daughter cells are formed. And in both the daughter cells, the number of chromosomes will remain same as that present in the parent cell or the mother cell, you can say. So suppose if the mother cell is diploid, because I hope the concept of diploid and haploid is clear to you, I'll just tell in brief. So suppose the parent cell is diploid, when it will form two daughter cells, both the daughter cells will be diploid. That means they will have the same number of chromosomes. Diploid means when the chromosomes are present in pairs. Suppose in case of human beings, we know there are 46 chromosomes, which can be written as 2 into 23. That means there are 23 pairs of such chromosomes. There is one pair, like this there are 23 pairs. That is called a diploid condition, D-I-P-L-O-I-D, diploid condition, where the chromosomes are in pairs. And when the chromosomes are only the single set, suppose out of the 23 pairs of chromosomes in human cell, if you take one chromosome from each pair, you will just get 23 chromosome. And when the chromosomes are in a single set, it is called haploid, H-A-P-L-O-I-D, haploid state. So diploid and haploid. So in mitosis what happens, the ploidy remains the same. If you begin with a diploid cell, both the daughter cells will have diploid number of chromosomes. And if you begin with a haploid cell, suppose N, then both the daughter cells will have haploid number of chromosomes. That's why it is called equational cell division, because the number of chromosomes remain same. So in any typical cell division, now this is the M phase that we were talking about. So in any kind of typical cell division, we have the following phases. First one is the prophase, second one is the metaphase, third one is the anaphase, and the fourth phase is telophase. These four are the phases of karyokinesis or nuclear division, which will be followed by your cytokinesis, that is the cellular division. So let us begin to understand what happens in the different phases. Suppose there is a normal cell, and in a normal cell, in the nucleus, you will not find chromosomes, rather you will find a network of thread-like structure which are called chromatin network or chromatin fibers. And this is what I have drawn here is the centrosome. There is an animal cell. In plant cells you don't have centrosome. There is an animal cell having centrosome. Each centrosome has two centrioles which are perpendicular in orientation to each other. Okay, so there is a centrosome. So there is a I phase nucleus. What is I phase? I showed you earlier. A cell which is undergoing G1, S and G2 phase. Now after I phase, when the cell will enter M phase, that means the cellular division phase. So first it will enter into prophase. Prophase, what happens in prophase? Slowly the nuclear membrane will start disappearing. The centrioles will start moving towards the two opposite poles, and as they start migrating towards the two opposite poles, between the centrioles you find that some fibers are being formed, and these fibers are spindle in shape. That's why they are called spindle fibers. And what happens to the chromatin fibers? The chromatin fibers starts condensing. Now as the chromatin fibers will start condensing, now they are regarded as chromosomes. So here I have shown one pair of chromosomes. Let's say there is a pair of homologous chromosomes. What are homologous chromosomes? You have got in class 10 that homologous chromosomes, suppose this chromosome, it is found in pair. So in a diploid condition, the chromosomes which are found in pairs, they are morphologically similar. Okay, they have similar alleles in homozygous at the same location, at the same loci. So suppose this is one chromosome and there is another chromosome. When we will call them homologous chromosome, when both will have similar morphology, they will occur in pair in a diploid state, and they will have similar alleles at the same location. For example, suppose here you have the gene for height, here also at the same location you will have the gene for height. Suppose here you have the gene for eye color, here also at the same position you will have the gene for eye color. So under such a condition, the chromosomes are called your homologous chromosomes. So here I have shown one pair of chromosomes, homologous chromosome. Just to show the difference, one I have drawn with black ink, another one is with red. So now as the cell will proceed towards late prophase, then what happens? The spindle fiber formation will be completed, okay, between the two centrioles. And these chromosomes, each chromosome, suppose there is one chromosome, each chromosome will split into two fragments longitudinally. They'll split into two parts, and each part is called a chromatid. So suppose the red chromosome, it will split longitudinally and it will form two chromatids. These are two chromatids belonging to the red color chromosome. And this, even the black color chromosome, that will also split longitudinally to form two chromatids. Now, so that is what happens during the prophase. The nuclear membrane will disappear, cell organelles will disappear, the chromatin fibers will start condensing, and after getting condensed they will split longitudinally to form two chromatids, and both the chromatids, they will remain separated, but they will remain joined at one portion, and the portion where they remain joined, that is called the centromer. Okay, so this point of joining, where they are joined, both the chromatids are joined, it is called centromer, centromere. Now next we come to the metaphase. So metaphase means it is the phase where the chromosomes, there is one chromosome, there is one chromosome, both the chromosomes will get attached to the spindle fiber. Now where they will get attached to the spindle fiber? They will get attached to the spindle fiber along the equatorial plane. These are the poles of the cells, and there is the equatorial plane. So along the equatorial plane, the chromosomes will get attached. So you can see to this spindle fiber, this chromosome has got attached, and at what point they will get attached to the spindle fiber? At the point of the centromer. Here also the red color chromosome has got attached at the point of the centromer to one of the spindle fiber. Now all the spindle fibers will not have chromosomes attached to them. Okay, only few spindle fibers will be used. So that happens in metaphase, that all the chromosomes, they align themselves in the metaphasic plate, or you can say along the equatorial plane. They get attached to the spindle fiber. From metaphase, now the cell will enter into the next phase, which is called the anaphase. What happens in anaphase? At the point of the centromer, it will split, and as it splits, the spindle fibers will start contracting. As the spindle fibers, they start contracting, both the chromatids, they start migrating towards the opposite ends. So you can see here both the chromatids have got separated. Here also both the chromatids have got separated, and they have started moving towards the opposite poles. As the anaphase proceeds, both the chromatids, they get totally separated from each other. This was one chromatid, this was one chromatid, this was one chromatid, this was one chromatid. But Here we need to understand one, you can say, nomenclature, because as soon as the chromatids separate out, now they are no longer known as chromatids. Now they are known as chromosomes. Is that clear? So till the chromatids were connected, they were regarded as, now you can say these are two chromatids belonging to the black color chromosome. Now here when the chromatids are getting separated from each other, they are known as individually, this is one chromosome, this is one chromosome. They are no longer called as chromatids. So that is the change in nomenclature which takes place once the chromatids get separated. And as they reach to the opposite ends, now during telophase, once again the nuclear membrane will start reappearing, nucleolus will start reappearing. The chromosomes, now they are called chromosomes, when they reach to the opposite poles, now they will start losing their thick rod like structure. Once again they will start becoming thread like structure, and along with telophase, simultaneously, side by side, there is no strict border, you can say. So with the advancement of telophase, the cell also starts dividing. Since it is an animal cell, the cell will start constricting in the center. There will be development of furrow, and this furrow will go on, you can say the cleavage will deepen out, and as the cleavage deepens, slowly both the cell membrane will separate out and we will get two separate cells. That is what happens in cytokinesis. Now there is slight difference between this cytokinesis of animal cell and plant cell. In plant cell, what happens is that, suppose there is the cell which will divide to form two other cells. Here what happens, from outside the cell will not constrict. Rather, from the central portion, the cell plate starts developing, and the cell plate starts elongating towards the periphery. So it moves towards the side. So it is more centrifugal in nature, outward seeking. But in case of animal cell, it is centripetal in nature. That means inside seeking. So that is the basic difference. Here from outside, the cleavage will develop towards inside. Here from the center, the cell plate will start developing towards both the side, and ultimately when the cell plate reaches the end, you end up getting two cells. So that is the basic difference between cytokinesis of animal cell and plant cell. So overall, these are the steps involved in mitosis. Now, what is the significance of mitosis? So one more thing we need to clarify or try to understand. So here we have seen how the chromosomes are getting separated. Now why it is called equational cell division, how the chromosome number remains same, I can show you here. We began with 2 chromosomes and we ended up with each cell having 2 chromosomes. That's why it is called equational cell division. To understand that better, and we know that chromosomes are nothing but DNA and histone protein. So how the DNA is getting distributed during the whole cycle, that also we need to clarify. So let us try to understand from here. Okay, so this is your distribution of chromosome and DNA in mitosis. So here I have shown 2 chromosomes. As I told you earlier, I have taken the example of one pair of homologous chromosome, and each chromosome we can say has one DNA molecule. This chromosome will also have one DNA molecule. So chromosome number is represented by n. So since there is a diploid condition, chromosomes are in pairs, so we have written twice n. DNA is an organic molecule, so we represent DNA molecule with C, so I have written it as twice C. It is in a diploid state. Now as this cell will pass through the S phase, I told you what is S phase, synthesis of DNA. What will happen? DNA amount will get doubled. So from this DNA, one more DNA will be formed, a copy will be formed. From this DNA, another DNA will be formed, a copy will be formed. So now the chromosome number remains twice n, but the DNA amount has become 4C. Okay. So after that, what happens? I told you that the chromosomes, they split longitudinally. As they split longitudinally, they will form chromatids. So this black color chromosome, it is splitting longitudinally to form 2 chromatids. The red color chromosome is splitting longitudinally to form 2 chromatids. Now the chromatids remain attached in the center, and this is the portion of what is called centromer. So Now in each chromatid we have one DNA molecule, and after that what happens, one more term you have to know. The chromatids belonging to the same chromosome are called sister chromatids. So these two are sister chromatids. Similarly, these two are sister chromatids. But chromatids belonging to two different chromosomes. Suppose there is one chromatid and there is another chromatid coming from two different chromosomes of a homologous chromosome pair. These are called non-sister chromatids. Now what happens, the chromatids will separate and migrate to two different cells. So from these two, let's say if I give them a number, suppose 1 dash and 2 dash. So let's say 1 comes over here and 1 dash goes over there. It's not fixed. 1 dash can also come here, 1 can also go there. That's totally depends on chance. Similarly, from these two chromatids, let's say 2 comes over here, 2 dash comes over here. So I told you, as soon as the chromatids separate out from the centromer region, they are regarded as chromosome. So till here it is twice n condition, because this is one chromosome consisting of two chromatids. This is one chromosome consisting of two chromatids. That's why twice n. DNA is 4C. But as soon as the chromatids they separate out, they are regarded as chromosome. So how many chromosomes we are having now? Twice 2 in pairs, so twice n, and the DNA amount also gets half twice C. So we began with twice n twice C. This was the mother cell, which ended up forming two daughter cells, twice n twice C, twice n twice C. So since the DNA amount and the chromosome number remains the same, it is called equational cell division. So what is the significance of this process? Significance is that this mitosis helps in increasing the cell number. So as we are growing from that single zygote, billions of cells have got added to our body. So all these cells have been formed by the process of mitosis. So it helps in growth as well as in repair. Suppose you get hurt, part of your skin peels off. So that skin will get repaired by the process of mitosis. New cells will be formed. So that is the significance of mitosis. It helps in growth and repair. Thank you.